Welcome to the Genovix Discovery tutorial. I'm Dr. Dieter Mulitz, the CEO and founder of Agronomix software, and I would be pleased to take a few minutes to give you a quick tour of Genovix, our all-new software for plant breeding and variety testing. So the interface has been designed to be quite easy to use and intuitive, like you would see maybe in Excel or Word with ribbon bars. So when you click on each main menu item at the top, it changes the items in the ribbon, and of course the icons that you can access for the tasks at hand, what you want to do. So here I have clicked to open up an experiment, and the first tab here tells me that it's happens to be an alpha design with 160 plots and some more details. And this software also, of course, does field plans. So here is included in the field plan. I can go to the whole field plan system, scroll up and down. This is what's in it, and there are many tools to design field plans. So let me go back to the actual experiment, like so. And if we look at traits here, it's very easy to add traits, and they would be defined as per the user's actual genotypes, your material, depending on the crop. And these are just some hypothetical ones that I've created. So in this system, we can transform data. There are many functions to do mathematical character transformations. We also have calculated traits. You can even do subsamples if you want to take data on individual plants. And analysis is fully supported. So I'll go back and just run these. So we have analysis by R scripts for more advanced biometrics, GLM, spatial analysis, and a lot of these are all built in the system. And our routines that we have developed provide extra possibilities for analysis. And if I go on to images, we can actually have images stored in the system. So here I have a photo for an entire experiment, if you like. And I'm going to look at Macoon as a variety, and this is an image that's stored for that genotype. So next we could have plot comments. Take a look at any comments that are stored for each plot, and those are kind of the main tools. If you want to look at some visualization, you can go here to chart type, and we have 3D surface and box plot and biplot. I'll take a look at contour plot. It's kind of an improvement over the heat map that a lot of people use. But we think this is kind of a better way of looking at the data to see if there are any trends in the field. And here's some high spots over here and so forth. Nothing dramatic like you might find in some fields with slopes and so forth. So this is one of many visualization tools that are available in our system. So those are the main tools here for the experiment. We also have reports and labels and so forth, export to Excel, and it goes on. Upon analysis, the means are stored in the database, for which you can build indexes and so forth. You can take a look at what was the ANOVA that was used to finish the analysis, and instantly it comes up and you have like a historical record. And of course I have seed allocation, like so. So here all the seed that was allocated tells me where it came from, what building, drawer, room, whatever the terminology you might want to use. And then simply a right click here shows a whole suite of tools for more functionality. I'll just go down and look at seed allocation report. And for people packing the seed, this is one of a number of types of lists available in Genovix to expedite the whole seed preparation and taking seed out of inventory and so forth. So those are just a few of the tools that are available just for experiments themselves. Next, we look at a breeding system that offers a lot of possibilities. Here I have the crossing matrix tool. I have a number of males and females chosen. Of course, I can flip those around if I want to do them the other way. And I have chosen a number of key traits, some marker traits for some alleles that I want present, and powdery mildew to see, let's say, some disease resistance, and some estimated long-term yield. So on that basis, the calculations are made. So this is one of a number of decision tools for the whole breeding system, and very helpful in the planning and designing process. So the other nodes, of course, are, let's say, for the crossing block. Once that's set up, there are extensive tools for all kinds of processes being made. I go to the breeding system here, and I look at a method. It could be any one of these sieve, cell, sterilization, CMS systems, and so forth, all supported within the system. And if we look here at advanced, in a nursery we're looking at advancing from F1, F2, F3, F4, could be self-selection and selection column and so forth. So all those tools are really there, as well as a number of special tools for creating hybrid seed, for hybrids, isolation nurseries and that, planting the females with the male, let's say, in the center, and then harvesting the seed and creating seed lots from that. So a lot of extensive tools, also line by tester and so forth. So a lot to explore within our system for plant breeding. Next on the research tree, we have genotypes and treatments, where you have all your germplasm data, could be treatments, parents, populations, hybrids, varieties, everything that's needed to really drive the system from creating process, setting up yield trials across many locations, and so forth. So here I have a third back cross. This is line conversion from B lines to A lines, and I'm following a pedigree system that denotes the number of back crosses built within. And the history tool allows you to just a few clicks take a look at the whole history of the development of the genotypes. If I scroll down here to something like YT, I can say, for this variety, where has it been planted? Well, exactly 112 experiments. Any click here takes me down to experiment, or if I like, I can click here on locations, and it will actually build a file with all the experiments and all the data everywhere that particular variety has been planted. In this case, it's 8,224 records, which is across years and locations, and it has all the other varieties planted with it. And I could easily build a filter here and just take out that variety. That's very easily done with some fairly standard tools in the Windows interface to find it. So pulling up the data, finding it is easily done, and we have a number of multivariate tools. So if I click here on, I have like a cluster test group, some simulated data, and if I'd like to look at clustering, for example, to maybe help decide on the grouping of parents for making crosses, along with let's say genomic prediction and so forth. So choose some traits under options, say, well, I would like maybe the break it down to the 10 cluster groups, and then we click cluster and invokes the algorithm, and we see some of the groups are quite different from others. This variety here is, for whatever reason, quite different from just about everybody else. So useful to know. Click on heat map and you get another visual representation here. You can easily see this is all stored in a matrix. So again, this is one of a number of powerful visualization and statistical tools available within Genovix. If we want to see things like Pedigree Connection, I have a genotype example here, and I'm going to go and say, well, right click, let's look at the dendrogram. Comes up like so, and just some random photos that I have assigned, and grab them all like so, and it brings them all over here, and all the data as well across the generations. So again, a way of really tying things together. Since this is a relational database, those things are very easily done, very much built into the entire system. As I said before, the traits can be assigned, named as you like with your own ontology or nomenclature. You can have things like percent, lodging, days to heading, number sign, so quite a bit of freedom to name things as you want. And with one click, I found out here I have 9,400 records of data for this particular trait, grain yield, as we see over here, and so forth. So I'm going to go to locations, look at Manitoba, all the information. Again, locations can have different traits. And if I look here at, let's say, Morden, click on the location icon here, the GPS, and it brings up our GPS tool, and it shows you precisely where Morden is to be found in the province of Manitoba. And let's go on to seed inventory, and I'll look, let's say, 80 YT Brandon, seed that was harvested there, put into seed lots and assigned, and quite a bit of flexibility and power to even go into anywhere in the building, cold room or whatever. So what do we have on hand at the moment? Let's say in this entire building, bring all the seed up, and you can decide on seed lot traits for the different species that you have to look after it. So, and then last thing could be like years, bring up all the data, look at everything, all the experiments in a certain year and so forth. So a very highly relational system, a lot of power and capability. So hope you've enjoyed this tutorial. Please contact us for a personal demo, find out information about Genovix and how it can empower your plant breeding, variety testing, agriculture research. Thank you.